Hello.
Good morning.
Uh, yeah, good morning. Hey, this is like the third or fourth day we're doing this, right? And it'd be nice to know if somebody's taking advantage of it. So if you're here in the comments, the live comments right there, say hello or what's up or how's it going or Guten Morgen, whatever floats your boat.
Christmas cookies.
Something. Let us know that you're here. In an effort to help people get done with the cell unit before break, so you have nothing to do over break, we've been live streaming and going through the different things that you had to do. Oh, look, and they're both mine. It's just, I'm sorry, Mrs. Smith. I know how competitive you are.
Listen, it's okay. Most of my students must be through the cell unit then, because why would you not show up if you weren't through the cell unit? So I guess they're done already.
There you go. So thank you, Hallie and Rosa, for coming in. All right, so let's do this. What we're doing today is running through part A, B, and C of the lab. We're going to do that really quick to kind of show you that. Tomorrow we'll finish the lab off. If you join us both days, my guess is you would have the lab done. If you've been joining for the other days, you should have those done. If you've been ignoring them, go back in the Blend announcements, click on the link, or just go to SOS Biology. You can still watch those videos and get the assignments done pretty easy. Oh, that was nice, Mrs. Smith. I appreciate your hand gestures there. All right. So in the lab, we're not going to read down through the whole lab. What we're going to do is show you the picture that you have to draw, talk about what you might have to label, and then jump and look at the analysis with you. All right, because a lot of the lab is how to make the slides, and obviously you can't do that from home, so we had to change that. The first part A deals with the cork cell, and I did grab, just because, like we spare no expense here. Right there is a piece of cork. All right. So, I want you to think about that piece of cork for a second. That's like you would put in the top of a bottle. Not that Mrs. Smith and I have a bottle, we just have the cork.
That goes to test tubes, actually. But go ahead.
Oh, okay. And you would scrape a little piece of this off and you would look at it. So the first thing you should ask yourself, is it alive right now? Just throwing that out there. All right. So jump into the lab. I think we start with a picture, right, Mrs. Smith?
Yeah, and Rosa, I hope we're back. I hope we're not still frozen.
Oh no, we froze on you. We certainly didn't do that on purpose. All right. How you want to do it? You want me to do the picture and then you can do the first set of questions?
Whatever you want to do.
All right, let's do that. Just because I'm already talking. Here we go. Picture of the cork cell. So we took a piece of this cork that I held up. We put it on the slide. We added water to it, put a cover slip under it, looked under the microscope, and this is basically what you would see. This is what you would see if you made a really, really, really good slide. And what you notice is we just have a whole bunch of little kind of circle-ish looking things. If anybody's in the chat, what are those? What would these be? What would these little circle-ish looking things be? Oh my gosh, if Hallie or Rosa answered, it would be almost more than my heart could take if somebody.
And I guess the thing that you should remember too is this is not just low power on the microscope. We're going to a higher power. We're going to a higher power. The higher power of the microscope.
The puns are bad today.
Oh my goodness.
Right. So we've really zoomed in. Any ideas with these little, I don't know. We got to remember they have a couple 30-second lag behind us too.
Right.
So
kind of circular-y type of shaped things. There's a whole bunch of them.
Stacked together. Okay. We're not going to wait too much longer. And it might be the lag. So what we're looking at is a bunch of different cells, right? And like I asked you, is this alive? Well, no. It's not alive anymore. It was once alive. So basically, there's only, what are you seeing? What's left? What's left after everything died? All the living material is gone. But this one cell part, which is a cell wall. So if you were drawing this and labeling it in a lab, really the only thing that you can label is the cell wall. So we see cells here, and we see the cell wall. And so now we jump over to the questions on the lab. So we go down to part A, because part A deals, oh, Rosa did rate cells. Yeah, you were exactly right. We go down to part A, and part A deals with the questions about what we just looked at. Mrs. Smith, if you want to hit those.
Right. So it says, is the cork you used alive? And so, like, at one point, cork, you know what I mean, is a plant and is alive. But when we're using the actual cork, no, it's not living. It's not going to get up and walk away off of something. It's not currently alive. And so the number 2, we kind of answered already. What are the small units that can be seen under high power? And so when you're looking under high power, we see those cells. And then we looked at the cells in there. It says, do these units appear filled or empty? Well, they kind of just look empty and they look clear. Again, because, you know, they are dead. I kind of think about like a skeleton. Okay, once a human remains or animal remains have started breaking down, like the skeleton stays for a lot longer. So the, you know, units are empty, but the specific part for number 4 that we still remain is kind of that outside support, which, you know, would be like the skeleton kind of, but are its cell wall. So that specific part that we still see is the cell wall that is left over. So I want you to kind of think about, when you're thinking about the cell membrane versus cell wall, what does that mean for plant versus animal cells potentially? And so then it goes into talking about Robert Hooke there and talks about, you know, these honeycomb units. So this is kind of repetitive from the first couple of questions. You know, what are the honeycomb units that he was looking at? Well, he's looking at cells. And again, what's the specific part that's left? Well, that's the cell wall. If there's cell wall, then is that cork a plant or animal? And then do animal cells have cell walls? And I kind of give you that answer as well. Okay, so cell wall only in a plant cell. And then the last one says, use your text to determine the name of a chemical which makes up the cell wall. So I know in my classes, I talked a little bit about this during the carbohydrate portion of our chemical basis of life. So it is a carbohydrate. It is a large carbohydrate. So it ends in ose. So if you think about cell and ose, you come up with the chemical that makes up some of the cell wall, or that's in there for that, and that is cellulose that we're looking at. So that doesn't make up the cell membrane, that makes up the cell wall, which is why, and we, in our bodies as humans, we don't break that down that well. So I kind of talked about that too.
Yeah, and that was actually a question on one of their note sheets, I think, too, back when we were doing that stuff. So they should have heard that word before. Cheek cells, next. So in this section of the lab, if we were doing it for real life in class, you would take a toothpick and scrape the inside of your cheek, rub the toothpick on the side, put some dye in to stain the cells, put the cover slip and water on it, and look at it. So the dye you're putting in, it's called methyl blue, and would stain the cells kind of a bluish shade. And so what you can see here on the screen is this bluish cell. So we have, I'm seeing 1, 2, 3, 4, 5, 6, 7 different cells maybe on here. And so let's just take this one right here, if you can see my mouse. If we scrape these out of our mouth and they're cheek cells, are they living or are they non-living? So they're probably still living. So they're also animal or plant. You scraped them out of your mouth. Let's go maybe animal. I see is a cell part that should jump out pretty easily to you. All right. So really, the only things that you could label, and I think the lab has you label some stuff. If I remember correctly, it's going to have you label the cell membrane around the outside of the cell here. It's going to have you label the nucleus, which is the darker spot. It's going to have you label the cytoplasm, which would be kind of that jelly-like filling of the cell, right? And I have, is this might be the one where they ask you to label the nuclear membrane, which is kind of weird, but is doable, I guess, if you would just draw your arrow to the very outside of the nucleus. I'm not sure if it's this one or not. One of them does ask for the nuclear membrane, which I always thought was kind of weird. So that's the cheek cell. That's what the cheek cell looks like. Hey, Elijah's here.
Hey, awesome. Elijah, we're going over the lab, the basic unit of life lab. So we're on the second.
Oh, wait, you need your, okay, so we need the questions.
Question.
If we jump to the questions, part B, here we go.
So this part, it's asking you to kind of describe the shape of a cheek cell. So when you're describing the shape of a cheek cell, it's kind of just asking you, you know, instead of the cork was that nice honeycomb type of shape. So a cheek cell is more rounded. So whether you put rounded, whether you put circular, oval, you know, as a teacher, I think we're just looking that you see that, you know, they have kind of more of a rounded shape because they don't have a cell wall. They only have that cell membrane. And that is because cheek cells, hopefully you know that you are an animal and not a plant. That would be good. So then, is a cell wall present? No, because again, you are not a plant. Mr. Shooter kind of talked about our cheek cells alive. You know what I mean? If we are doing the lab and scraping them and doing it right then and there, then yes, you know, those would still be alive. And I know we couldn't do that, unfortunately. But yes, they would still be alive. And then it's asking you to describe the location of the cell membrane. Okay, that you labeled it on there. Just think about that's around the outside of the cell. It says use your text, but the cell membrane, we talked about that in the live stream the other day. And again, if you haven't watched those live streams for the past couple of days, go back in your Blend announcements, click on them. We go through all those documents that we're doing. And so like, look at that, back to that function of the cell membrane, but it's basically just supporting the cell as well as regulating what comes in and out of the cell. The cytoplasm, again, this is from that other paper. Cytoplasm is inside of the cell. The cytoplasm is a jelly-like substance, mainly made of water. It's going to be a good conductor, which is good for metabolic reactions, and it is also going to be, used to, kind of keep the cell together. Mr. Shooter, if you missed it, said at the number 8, the stain added to cheek cells, that would be methyl blue. Okay, it's a blue dye. There's other things we can add. Methyl blue works really well with animal cells. And then the last one, do you have evidence that living things or once living things are composed of basic units called cells? The answer would be yes. And explanation, there's this cool thing that we've talked about called the cell theory that, kind of supports that. So
Not to mention the last two things we've looked at have been made of cells.
Correct. And like I said, I see a couple of my students popping in here and everything. So
yeah, I mean, it's nice. Now we have 2 of mine and 2 of yours. We need time.
People getting work done. Like to see it.
All right, so let's keep going. We want to do part C. We want to keep these as kind of short as we can, but get through the stuff for you. So we thought today we would break this into part A, B, and C. And then tomorrow we'll do the other 2 parts of the lab. And so part C is the cell nucleus and nucleolus. So we are looking at a picture here of an onion cell. So what we would have done in lab if we were able to do this, sad face, we would have taken a piece of onion and got a really, really thin layer of the onion off, which is really cool. You can do that. Put it on the slide. We would have added some stain to it to make it so we could see it. This time we would have used iodine. Put some water on it and a cover slip. and then looked at it under the microscope, and this is what we would have seen. Now, there's a hint. Like if I didn't tell you this was onion, you should look at this and right away go, hey, that's probably a plant. And that thing, these cell walls, right? Because we can see cell walls. People crack me up when they draw this. When I mean, look at it. Here's a cell, right? Here's a cell. Wait, you can't see my mouse because I'm drawing on the wrong screen. Here's a cell, right? It's not just a square or a perfect rectangle. It really isn't. But I mean, we're building a wall here. It's almost like bricks in a wall. It's just another brick in the wall, actually. That's for you, Mrs. Smith. And then we see these kind of darker purple. They look purple on this screen anyway. Those would be our nucleuses or our nuclei. And then on my screen, I don't know how this will go over YouTube, I can see like a little dark dot in the middle of the nucleus. That would be your nucleolus. Sorry. Oh, boy. All right, so what could we label? We could label the cell wall. We could label the nucleus, maybe the nucleolus. Of course, you could label the cytoplasm. We know we might not be able to see it, but we intuitively know right inside that cell wall is a cell membrane. And we know around the nucleus there's a nuclear membrane too. And I think that about sums up what we need to know from that picture.
Before you switch to Mr. Suter, will you go back real quick? So like if you look and you said like here's the cytoplasm you're pointing at, that you're looking at that picture, scroll back up to the cork for a second and see the difference between one that's filled and one that's empty. When we talked about are they empty? You know what I mean? Like you can see that on there from looking under the microscope. Empty, nothing there. And then the filled, there's the nucleus, and you can even see that shadow of the cytoplasm. Okay, questions. Sorry.
Good. All right, so questions. We're almost there, guys.
As long as you're watching Elijah, that's all that matters. Don't worry about where you're watching or how you're watching it.
Here we go. Last set of questions for today.
So describing the shape of an onion cell. So again, not a perfect rectangle, but kind of rectangular in shape. Are onion cells produced by plant or animals? Well, Mr. Suter said plant. Is the cell wall present? Well, if it's a plant, the answer is yes. The shape of the nucleus of an onion cell, more circular. Within what cell part already studied does the nucleus lie? So like the nucleus is within the cytoplasm inside of the cell membrane and cell wall. The nucleus controls all of the reactions in the cell. And it also holds the genetic information. Again, it says consult the text, but like we explained all of that in the live streams the last couple of days. Describe the shape of the nucleolus of an onion cell and where is that found. So that was that little dark spot in the very center of, or not very center, but in the center of the nucleus. And the function, like Mr. Suter talked about the other day, is that it is responsible for producing ribosomes. And then the last part there, what structure separates the contents of the nucleus from the cytoplasm? And that would be the nuclear membrane. And why were the cells stained? And so the cells are stained to make it easier to look at underneath the microscope. If you don't stain your cells, everything kind of looks clear. You can't really see everything well. And the nucleus definitely isn't dark like that. So the stain really helps us to see things for it to be visible underneath of the microscope.
I was wondering.
Yes, please come tomorrow, Rosa. We're going to finish it. And just like we've done, at least I'm going to do, if you show up to both sessions, that's your lab. You'll be done, at least for me. So I'm speaking for Mr. Suter quickly because Rosa had to go.
Okay. Yeah, I think that's it. Hey, once again. So just a couple reminders. If you're submitting things in Moodle, if you're putting links to your Google Docs or whatever in Moodle, you need to share them with your teachers too. Like we can't see it when we click on it if you didn't share it with us. So there's a lot of people that are struggling with that. Trying to finish the cell unit before break so you have nothing to do over break. But definitely finishing it before you come back from break. And we'll see you tomorrow for part D and E, right?
Mm-hmm.
Trying to make it easy for you guys.
Yes. And Right. And my students that are here, like if you're sticking around both days, like we're going through everything. Like you will be done this lab.
So
The lab was due weeks ago.
big shout outs to Anthony, Rosa, Elijah, and how do we say
Oh,
it?
how do you say her name? It's
Hallie. We love messing with you, Hallie. All right, so have a great day. That's it, right? Anything else?
yeah. Reach out to me if you need anything.
All right. Yeah. Later.
Later.